Hello and welcome everyone to this podcast on endocytosis. We've already talked a little bit about endocytosis, but let's remind you of what a working definition could be. This is a pathway that brings material into the cell through the invagination of the plasma membrane. We can think of endocytosis falling into one of three categories. The first is pinocytosis. We can think of this as cellular drinking. It'll bring in small amounts of fluid and molecules. The size of the molecule is going to be less than 150 nanometers. It is also nonspecific. It will bring in whatever happens to be here as the vesicle is pinched off. A few other things about pinocytosis. All eukaryotes do this. Pinocytosis, as it's bringing in these vesicles, you can see that we're also losing membrane. So it's fairly well balanced with the secretory pathway so that the overall size of the cell doesn't change much. So depending on which cell type you're talking about, the rate at which these vesicles form through pinocytosis can vary. But we typically say that this is continuous. It may be slower or faster depending on the cell type you're talking about, but it's occurring at irregular intervals. Let's talk about the second pathway now. And so let's talk about phagocytosis. Just like pinocytosis was equivalent to cellular drinking, phagocytosis is thought of as cellular eating. Pinocytosis brought in molecules that were less than 150 nanometers. Phagocytosis brings in particles that are greater than 750 nanometers. Now, not all eukaryotic cells undergo phagocytosis, because phagocytosis requires specialized cells. Some cells that undergo phagocytosis include protozoans. They will bring in bacteria, and they do this for food. That's how they eat. In animal cells, they use phagocytosis to bring in bacteria, but it's for defense purposes. The way that most animals will obtain their food is they will release digestive enzymes into the lumen of the digestive tract to break down the food particles into much smaller particles that can diffuse across the membrane or be transported across the membrane using one of the active transport pathways we've previously talked about. In animals, there are two main kinds of phagocytic cells, and I'm going to draw them over here: neutrophils and macrophages. A few differences between neutrophils and macrophages. Macrophages are larger and longer lived, so neutrophils are smaller and shorter lived. Neutrophils are typically first to the site of infection, whereas macrophages are late to infection sites. They are both part of the immune system. They are both considered first responding cells to sites of infection. A cellular difference between these, and I'll write it up here, is that neutrophils contain a lobed nucleus. Their nucleus contains 2 to several more lobes on it. A macrophage has the typical round nucleus. So that's a way that you could tell them apart if you were looking at two cells that appeared similar. I'm going to erase this part here that I just wrote because I need some room here to draw what's happening during phagocytosis. Okay, so let me draw out how it might look if a macrophage or a protozoan were to engulf a bacterium. You're going to have your macrophage here. It could be one of the other cells, but let's go with macrophage. And what happens is the bacterium will interact with this macrophage in such a way that it stimulates the invagination of the membrane, where it's going to pull this bacterium in. So it'll slowly begin to wrap its arms, so to speak, around it. And these, I shouldn't call them arms, I should call them what they are: pseudopods. And they will slowly move around the bacterium. And the way they're moving around, and we'll talk more about the cytoskeleton later, but these cytoskeletal elements begin to organize in such a way that will push the membrane around the bacterium until they get to a point about here. And at this point, these final tips here fuse. And when they fuse together, these two will fuse, and these two will fuse. And what will happen is you'll have this vesicle inside of the macrophage. And the vesicle inside of the macrophage will now have the bacteria in it. And then the plasma membrane will refuse and stay intact. This structure here that we just drew, this engulfed bacterium, and it wouldn't have to be a bacterium, whatever has been engulfed by the phagocytic cell, we call a phagosome. Now what happens to this phagosome? I'm going to make this cell a little bit bigger so we can draw everything that's going to happen here. The phagosome will now fuse with an endosome. So this is an endosome. It is then transferred and fuses with a lysosome. And we'll talk about lysosomes a little bit later in a little bit more detail. Inside of here, the bacterium will now be digested. And all the material in it can now be used. Either if it's a protozoan for food, or if it's an animal, it will have gotten rid of that infectious agent, as well as recycled the material from the bacterium. As you can see, the fire alarm is going off. So I'm going to stop this podcast for a moment, and we'll pick this up in a bit. Okay, luckily the fire apparently is out, and so we can continue with the podcast. I'm sure you were all quite worried, but let's move on to our third type of endocytosis. And this is called receptor mediated endocytosis. So let's talk a little bit about the receptor-mediated endocytosis now. It's similar to pinocytosis in that it brings in small amounts of liquids and small molecules. About the same size limits as well, less than 150 nanometers. But what makes it different than pinocytosis is that pinocytosis happens to bring in any molecule, as shown here, that happens just to get trapped as the vesicle forms. This brings in molecules specific to a receptor it binds to. So remember, this is what we've already talked about with clathrin-coated pits, where we have the receptors here that bind to a specific molecule. And let's be real specific here. Let's say it's going to bind to a molecule called LDL. Now, LDL is a molecule that is in our blood that will hold cholesterol. So attached to this LDL is a molecule of cholesterol. And as long as we have typical levels of LDL in our blood, it will bind up the cholesterol and it'll bring it into the cells. So LDL is going to bind to these receptors. And we're not going to go through all the steps that we talked about before, but you will eventually form this clathrin-coated vesicle that has these LDL receptors that is bound to LDL. And remember, at the early stages, it's going to have those adaptins and the clathrin coats on it, which I'm just going to signify with this one line here. I should draw the clathrin and adaptins here too. And then it loses the clathrin and adaptins, and then just has this vesicle with these receptors on it. And of course with the LDL. Now what will happen is this will fuse with an endosome. And so we'll have a larger vesicle here with the fused vesicle and the endosome. And in this endosome, the receptors will release the cargo. And in this case, they're releasing the LDL that contains the cholesterol in it. This then will fuse with a lysosome. And then in this lysosome here, the LDL is degraded, which I'll just show with an X going through them. And as it degrades it, it releases cholesterol, which can be used by the cell in various pathways. Remember earlier in the semester we talked about how this can become a component of the membrane, which is helpful in maintaining a constant fluidity of the membrane. So that occurs here in this lysosome. Now you may notice when I drew this over here, I didn't draw the receptors. And that's because what happens here is these receptors will pinch off of this endosome and form a new vesicle that contains the receptors. I lost my color coding here. I should have made these green. Again, they don't have the cargo anymore because they lost it. But now what will happen is that this vesicle will refuse with this membrane here with the intention of releasing those receptors back to the membrane so that they can be used in the next round to bring in more LDL cholesterol. Now I'd like to spend a little bit of time talking about the lysosome here. Okay, so the lysosome. It's an appropriate time to spend a little bit of time talking about the lysosome because we've just talked about its role in receptor-mediated endocytosis. So let's draw our lysosome. The lysosome contains approximately 40 hydrolytic enzymes. They will digest a variety of molecules, almost anything that it comes in contact with, but certainly DNA, protein, RNA, lipids, and sugars of various sizes. Interestingly, the pH of the lysosome is about 5. So it's a very acidic environment. The pH of the cytosol is about 7.2. This pH is very important because lysosome enzymes only work at a low pH, like 5. This is very helpful in many ways. One, we are only allowing digestion of these molecules in the lysosome. If something were to happen and the lysosome would break up and these enzymes were released in the cytosol, the pH of the cytosol would prevent those enzymes from working. So you wouldn't have the entire cell being digested, which on most days is a good thing. Now, how does it maintain this low pH? It maintains this low pH by having a proton pump. The proton pump has ATPase activity where it takes an ATP, hydrolyzes it to make ADP. In that process, it will move protons into the lumen of the lysosome, keeping that pH at a low pH. Now, as I said, we have about 40 enzymes here in the lysosome, as well as some other proteins. And all of the proteins, which includes the enzymes, enter the lysosome in a vesicle from the ER, or from the secretory pathway, so it goes from the ER to the Golgi to the lysosome. There is one term that we haven't mentioned yet that we need to mention, and it's linked to the lysosome, so it's appropriate to mention it now, and that is autophagy. We don't have much time to spend on it, but I think it's important to at least define it because it's a term that may come up in a class as you move forward with your education here or at another school as you move on, and it may also be on the exam, so it's an important word to know. Autophagy is when the lysosome digests obsolete cell parts. In a real sense, this is a pathway using autophagy where the cell eats itself. As parts of the cell need to be recycled, organelles need to be recycled, or during cell death, these parts of the cells need to be digested. Okay, that ends this relatively short podcast on phagocytosis. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.